Welcome viewers. Today we are going to see the topic industrial enzymes. Here we will be covering briefly about enzymes, their activity, and then learn the different industrial enzymes in use. We will also see briefly about the production processes of three different industrial enzymes, that is amylase, protease and cellulase. What are enzymes? These are biological catalysts which speed up a chemical reaction within a cell and itself does not get changed. These are meant for proper functioning of the biochemical activities within a cell. There are several operations. How they do is like either they can cut, that is they may degrade like this, two compounds are similar broken down into two separate ones. They may build or used in synthesis, that is two simple compounds may be joined or may be also involved in modification. You can see here a smaller compound being modified to a larger one. So basically all enzymes operate with the substrate. They are very specific to the substrate with which they will act and bring about certain defined changes. The sources of enzymes are basically plants. We have examples of papain from papaya. We have also pineapple as a source which generates bromelain as an enzyme. We have animal sources which are typically the cattle or typically cow which produces a rennin used for cheese making. And most importantly, we can see here 80% of all the enzymes produced are derived from microorganisms. There are limitations of deriving enzymes from plants and animal sources, and we can see here how we can actually produce such from different sources. If we use animal or plant material, we'll go for a grinding step, then the extraction of the enzyme from that source. While if we are using microorganisms, we will carry out a fermentation protocol in a fermenter, and then depending on whether the enzyme produced is intracellular or extracellular, carry out different protocols to get the final product. The final recovery involves filtration, concentration, purification, drying, and there may be several others also like chromatography, dialysis and many more. All these makes the final product slightly expensive. But we are generally faced with problems if we are using plant or animal sources. They generally produce a combined source and different varieties of enzymes are obtained all at once. To get purer enzymes, microorganisms are the best choice. Now we will briefly venture into the detailed protocol of the downstreaming, that means purification step. If the enzyme is extracellularly derived, then it is easier. We can go directly to the filtration step. While if it is intracellular, the cell first needs to be broken down. That is often done by simple grinding or at times by ultrasonication. The filtration process generates some cell biomass which is used as some products like animal feed, but the broth contains the mainly enzyme in solution. This enzyme in solution can be differently downstreamed, like it can be concentrated by evaporation or several other processes like osmosis. One typical example is that of protease in the chemical industry. While you can carry out precipitation of this particular component to generate a crude enzyme which is pectinase used for fruit and other vegetable pulping and juice or other related manufacturing industries. And we can also carry out a chromatography which will generate glucose oxidase. Now this chromatography generates purer compound and that is used in the medicines. Now enzymes are present in all cells, human, plants, microorganisms and several. In the human being, basically amylases, proteases and lipases are important, and these are produced by the cells themselves. Whenever there is a lacking or there is requirement from external sources, we require it from the industrial processes that enzymes be produced in the pure form and then taken in the form of medication. These are the two plant sources of enzymes. Now basically the human body requires amylases, proteases and lipases for digestion of the food that they consume. But we will be looking into some other categories of enzymes, not with the respect to human need but the industrial production, because industries like cloth and paper and likewise dyeing manufacturing processes, the bakery, breweries all requires enzyme, and that is why huge amount of such enzymes are produced, and they include amylases, proteases and cellulases. First, we will start with amylase. This is a particular type of enzyme which cleaves the starch molecule into glucose units. Starch is a polymer of glucose, and there are two types. The unbranched starch is called amylose. We can see the unbranched here, and the branched chains are known as amylopectins. These two comprise the starch. We can see here again the glucose units of amylose and the branch chain structure of the amylopectin. Now, amylases will act on starch, and that is why there are several types of amylases. We have alpha amylase, beta amylase, glucoamylase, the common three types. Then there are others like the isoamylase. Let us see how they work on. This is the straight chain amylose and this is the branch chain amylopectin. Alpha amylase generally acts in the endo position, that is internal. So wherever there are the straight chain glucose units, the endo acting alpha amylase will work on it. This red arrow shows it. Here also in the branch chain, wherever there are single linear type of glucose units, they will be cleaving it. The glucoamylase on the other hand will be cleaving here, you can see from the non-reducing ends, while the beta amylase will be involved in cleaving two glucose units. From here also you can see here, from here also two single glucose units will be cleaved off together called as maltose, while the isoamylase generally acts on the branch points. Now we move on to the organisms which produce amylase used in the industries. They are Bacillus licheniformis, Bacillus amyloliquefaciens and Aspergillus niger in the fungal category. Industrial alpha amylase production can be of two different types based on the source. We can use bacteria, so that will generate bacterial alpha amylases, and we can also use fungi as a source to generate fungal alpha amylases. These are more important. They generate more of the purer types of compounds and are easier to produce. And we see that mostly they are produced by submerged fermentation and less by solid state. While on the other hand, if we are using fungal alpha amylases, they are produced mainly by solid state fermentation and less by submerged fermentation. Briefly, we will look into the media composition required for production of alpha amylase. This interestingly includes starch. Now, starch is a complex polymer as we have already seen, and this is the inducer required in the medium so that the organism of choice, that is Bacillus subtilis, produces the amylase. Generally, they may not produce excess amount of amylase in the medium, but we have to induce the cells by adding starch. Starch is a large macromolecule and often cannot enter the cell directly. It is considered that the amylase enzymes are constitutively produced by the cells and they are excreted outside. These will degrade the starch present outside in the medium to smaller components, as we have already seen, of glucose units, and these then will act as an inducer. Now, we have seen briefly the production, how it happens in the industries. As we mentioned, submerged process is the most important one for bacterial alpha amylases. Amylases generally are produced best at 45 degrees centigrade, but most often the temperature is kept slightly less, that is around 35 to 37. This generates more amount of amylase. And likewise, we will see the application now where they are applied. Several industries utilizes amylase. And the first is obviously maltose. As we have seen, two units of glucose together are called as maltose, and that is generated mainly for the beer producing industries, and the amylases are involved there. Then there is a bread production industry or the baking industry, which also requires amylases. Then the beer producing industry obviously works with barley and then malt, so that also requires amylases. We have several others like the plastic candy, and many other foods like MSGs also require amylases. Now we will move on to the second category of enzymes, that is protease. In the category of protease, we will see that mostly in the detergent industry we will find the application of proteases. Proteases are enzymes which actually degrade protein into their respective amino acids. They are involved in cleaving the peptide bonds between the amino acids and release of the amino acids. They can be classified into several types. The first classification type is based on the nature of the component that is present in the enzymes. The serine proteases essentially have serine, like example chymotrypsin, trypsin. Cysteine proteases are mainly this papain and mammalian cathepsins. Aspartyl proteases contains aspartyl moieties, and the example is rennin, which we are very much useful in the food industry. And metalloproteases are the ones which contain some type of proteins along with metals. But we are more interestingly concerned with the classification scheme which applies with respect to the different pH types. The first among them is the alkaline protease, the second neutral, and the third acid, depending on the pH. The alkaline proteases are the most important and often used in the detergent industry as small particulate matter with different colorations like green or blue. These are the ones which help in the removal of the stains, releasing the amino acids from the protein components. The neutral proteases are quite unstable but used mainly in leather and food industry, and the producer organisms are Bacillus subtilis, Bacillus cereus, and here we can see the diagram of the fungi Aspergillus oryzae, which also produces the neutral proteases. And in the acid protease range, we have a typical example of rennin, and we know that rennin is an enzyme used for the preparation of cheese. Now we will briefly see the alkaline protease production. Commonly, the marketable or the trade name of all these alkaline proteases produced from Bacillus species are bacillopeptidases, and one among them is the subtilese in Carlsberg produced from Bacillus subtilis. This is the structure of the protein structure of subtilese in Carlsberg, and the source organism is Bacillus subtilis. If we look into the common processing steps, we will see that sterilization is a very important step during the production of protease. Other organisms contaminating will actually reduce the yield of protease. Organisms may be in the lyophilized condition or in the nitrogen, liquid nitrogen storage. Taken from there, we can grow them in shaken flasks and fermenters at 30 to 37 degrees centigrade. Then we can incorporate into huge amount of fermenters like the 40 to 100 cubic meter size. Fed batch process is often employed not only for protease but also for other enzymes, because most often the enzyme production rates are slower if all the glucose or the carbon substrate are fed at once. That is, catabolite repression is a major problem here. Aeration must be maintained at approximately 1 VVM, and the time required for the entire production is 48 to 72 hours. We can see here the carbon source, which is either glucose or starch, and the nitrogen sources are casein, yeast extract, peptone, tryptone. This is a typical pictorial representation, that is, in the shaken flask, first we will grow the lyophilized cultures. Then we will have a pre-seeding tank, that is increase of the amount, and then go for the seeding fermentation where the organisms will be grown in huge amount. That will be incorporated as inoculum into the industrial fermenter where the production medium is present to generate the proteases. And downstream or harvesting, we have already discussed, requires chromatography, precipitation, filtration and other processes. And we will briefly mention about rennin here as required by the cheese industry in huge amount. Rennin is used for cleaving the casein moiety into different components that generates the curdling process and gives rise to the cheese that we can so happily enjoy. Now we are moving into cellulase. Cellulase is a different kind of enzyme specially required for the degradation of the plant material which contains the cellulose and hemicelluloses. Now cellulose was obtained from the plant material, as we can see, and any plant material which is woody in sense may have the three components of lignin, cellulose and hemicellulose. Now cellulose, if we see the structure, it is a linear polymer of beta 1,4-D glucopyranose units linked in this fashion. And the enzyme cellulase actually is an enzyme system, means it has three different or more different types of enzymes which are working in coordination to generate or cleave the cellulose into its respective units. and these components are endo-beta-glucanase, exo-beta-glucanase and beta-glucosidase or cellulase. How they act? Now, if we can look into this diagram, we will see these endoglucanases are acting on the internal positions of the polymer, while the exoglucanases are acting on the outer sides. And these beta-glucosidases are then cleaving off the free molecules of these simpler chains into free glucose units. The whole thing is represented again with the actual structure of the polymer. The cellulose crystals are broken down first by the endocellulases or endoglucanases, and then that exoglucanases or exocellulases are acting on it to generate these cellobiose, which are then cleaved by the beta-glucosidases to generate free glucose units. Now, a complex polymer like starch, if can be broken down into simple glucose units, it will be easier for several microorganisms to utilize, and that is why required in several different industries. Once such production is shown here, that is cellulase production by Acremonium cellulolyticus organism. Now, here we will see that the production takes place in a huge fermenter at 28 degrees centigrade. The organism is inoculated and paper sludge is also used here as the medium. Once the production happens, then there is the separation of solid liquid and the broth containing the cellulases is taken to another bioreactor. The solids are used for several other purposes, while the broth which contains the cellulases used in the second bioreactor are inoculated with Saccharomyces cerevisiae. These are yeasts which will be employed for the production of ethanol. So, this generates ethanol, and the cellulase enzymes are used for the saccharification of the paper sludge, which is used as the medium again. So, we see here that cellulases can be derived at one site as well as can be utilized on the same site using a second bioreactor. Same type of thing can be done with filamentous fungi, which are used as the producer organism for these cellulases like the trichoderma. We move on to see where these cellulases finds their application. Basically, they are used for the preparation of washing detergents, in fiber modification, several pre-treatments of pulp, the de-inking process from used papers, additive for livestock, additive for food, and we will be seeing how this biofuel is getting produced. We have just seen that cellulases are used for the production of ethanol. So, ethanol when blended with the petrol or diesel gives rise to biofuel, can be used by the vehicles. Now, this is an important criteria here because we are having or facing the problem of petrol and diesel shortages, and that is why the biofuel is the future fuel for us. And the most amount of plant material which are degrading or lying just like that, containing lignocellulosic or cellulosic materials, can be easily used for the production of ethanol, which can be used for fuel. The sources should be cheap, and that is why the straw and other material containing celluloses are used upon by using cellulases from Trichoderma or Aspergillus species, and then ethanol can be produced. So, after seeing all this, we can say that none of the industries that we generally cater to, like the food industry, leather, paper, pulp, textile, the medicines, all require enzymes, and we are indebted to the biotechnology these days that have generated processes from different microorganisms to generate these enzymes, which are severally used and required by all of us.